Very warm good morning to one and all. Present in this countrywide classroom building program 18 days, organized by Faculty of Science and IPC of Digambar Bindu Arts, Commerce and Science College. Dear friend, first of all, yes, I am audible, ma'am.
Yeah, sir. Perfectly audible.
First of all, I'm apologies for all the who joined this meeting and our resource person Aditi Deshpande, madam, ma'am. Uh, as due to because of poor connectivity, I am not able to connect with connect in this meeting earlier. But now, without delaying this session, today's 18 days morning session, we are going to start here. And the resource person for today's morning session of 18 days of countrywide classroom teaching program is Miss Aditi Deshpande. She is a microbiologist, and topic for discussion in countrywide classroom teaching program is the prokaryotic DNA and its replication. Now I handed over, firstly I welcome Aditi Deshpande madam and handed over this session to Aditi Deshpande ma'am. Aditi Deshpande ma'am, session is over to you.
Yeah, sir. Yeah. Thank you, sir. Let me first of all start my expressing profound and most sincere gratitude to you, all of you, for honoring me your invitation to me. And at the outset, I wish to thank Honorable Chairman Dr. DB College, who explored his views through this long classroom program. Right, I humbled by this kind gesture in giving invitation to me. I wish to assure you that I will be always at services to your college, definitely. I take opportunity to thank Honorable Principal Dr. Punjab Raoji Chauhan sir, coordinator and convener of CWCP 2021, Dr. Arvind Chauhan sir, and the other esteemed members of DB College, and all the beloved students and present over here. I appreciate your love and sense of devotion in the facilitation of the students. Uh, Please permit me to acknowledge with thanks all those for their worthy donation of time and invaluable support during this conference. Thank you. But yeah, before start my lecture, I please beg apology because of my my sound disorder due to throat infection. So please now I will start my lecture. Thank you. Now, there are actually some contents like it includes introduction, basic terminologies, basic fundamentals of DNA replication, and what exactly the prokaryotic DNA replication is. Now, exactly the basic terminology is what is molecular biology. So, molecular biology is the study of biology at a molecular level. The field overlaps particularly with other areas of biology and chemistry. It studies on DNA, RNA, and functional, that is, proteins, and the macromolecules involved in genetic information and cell functioning. It contains central dogma to study the genetic information. As in this diagram, we can see like there is a gene in the right bottom side, like, so this is a molecular biology. From that side, this is a molecular biology initiates. From the genes, it it's transcribed into RNA and it's translated into proteins. So there is a protocol like there is a protocol of molecular biology. It is also known as central dogma of molecular biology. The central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product or a protein. The process by which the DNA instructions are converted into the functional products is called gene expression. In this diagram, you can see a two-stranded DNA. Okay, in left side, blue colored, two-stranded DNA. When the two-stranded DNA forms its original copy or like template copy, it's also known as DNA replication. We will see step by step for furthermore. And then when DNA forms RNA, the process is known as transcription. When the RNA translated into a functional protein, it is known as translation mechanism. And this all continuously known as molecular dynamics. Now the basic structure of DNA. DNA is a deoxyribose nucleic acid. It is a polymer of deoxynucleotides having usually double-stranded structure. It acts as a genetic material in most of the organism, which carries the genetic information. DNA has three main components like deoxyribose, which is also known as pentose sugar. Second content is base. There are four bases like ATGC, adenine, guanine, thymine, and cytosine. And third one is phosphate. From the phosphate, it forms the new chain. Okay. Now the basic structure of DNA. DNA is the whole package of pentose sugar, nitrogen base, and phosphate group, which forms one nucleotide. In this image, you can see how the one nucleotide is formed. One nucleotide is formed by one phosphate group, then nitrogen bases, ATGC, and pentose sugar. Now, what is DNA replication? It is the biochemical process that involves the synthesis of new DNA molecule of mirror image of an DNA. DNA replication occurs in all living organisms, acting as the most essential part for biological inheritance. DNA replication occurs in cytoplasm of prokaryotes and in the nucleus of eukaryotes. DNA is replicated during the S phase, like synthesis phase, which is the first interphase of our cell cycle. Now, how DNA replication happens, we will see. A reaction in which daughter DNAs are synthesized using the parental DNAs as template. You can see in the diagram, in the left side, there is a parental DNA, okay, and there are two strands in red color, which is represented by red color. And how exactly replication occurs? When the parental DNA, when there are two strands of parental DNA, replication means what? When those parental DNA is utilized by daughter DNA just for template side. Okay, so this is known as replication. Transferring the genetic information to the descendant generation with a high fidelity. There are actual three possible replication patterns. One is semi-conservative replication, second one is conservative replication, and third one is dispersive replication. So what exactly the conservative replication is? So it would leave the two original template DNA strands together in a double helix and would produce a copy composed of two new strands. Okay, in this diagram you can see one parental DNA. Okay, one parental DNA having its own copy. Okay, and its daughter DNA having its own copy. So conservative it would leave the two original template DNA strands together in a double helix and would produce a copy which composed of two new strands containing all of the new DNA bases pairs. Now in semi-conservative, in all of the prokaryote and eukaryotic organisms or cells, their semi-conservative replication mostly occurs. So what exactly semi-conservative replication, we will see. Like in this type of replication, it describes the mechanism of DNA replication in all known cells, as we have discussed earlier, in which double stranded molecule of nucleic acid, which separate into two strands. In this diagram, you can see the two strands get separated, and upon which new strand is generating its own one. Okay, so parental strand is using to develop new strand, and which would be a complementary to the parental strand. Now in the third image, you can see this is a dispersive model or dispersive replication. Actually, this is here original DNA chain breaks in random wise fashion. Okay, so we cannot say like this would be in same fashion or different fashion. It must be in a random fashion. Then characteristics of replication. There are four characteristics of replication. First is semi-conservative replication. Another is bidirectional replication. Third one is semi-continuous replication. And fourth one is high fidelity. Now, the direction of replication. Actually, in prokaryotes and in eukaryotes, there is a bidirectional replication. In eukaryotes, there is a one way, or like in some cases it would be in one direction, or in some cases it would be in a bidirection. So now what exactly the bidirectional replication is? Replication starts from unwinding the double-stranded DNA at a particular point, which is also known as origin of replication. Okay, also known as origin or ori site, followed by the synthesis on each strand. The parental double-stranded DNA and two newly formed double-stranded DNA form a Y-shaped. Okay, we will see further in the upcoming slides. You will get it soon. And Y-shaped structure called replication fork. Now, there is a Y-like structure. Okay, so first is 3 prime. This is 5 prime to 3 prime direction. Okay, so this is a leading strand. Leading strand means a new nucleotide gets added in the 5 prime to 3 prime direction. Okay, in lagging strand there is some gaps. We will see. You will get it soon. Okay, so this is a Y-like structure. Just I want to say that. I want to show that. Now, what exactly the prokaryotic DNA replication is? Prokaryotic DNA replication is the process by which a prokaryotic duplicates its DNA into another copy that is passed onto daughter cells. Replication, actually, prokaryotic replication is bidirectional and which originates from a single origin of replication point, which is also known as ORIC or origin of replication site. Although it is often studied in the model organism, which is generally in prokaryotic, we use generally E. coli, Escherichia coli for further studies. So here we have taken E. coli bacteria for our study. Okay, now it is named theta replication. We will see in the upcoming slides what exactly the theta replication is. Okay, now there is a theta replication type. In this image, you can see a theta-like structure of DNA, or how it replicates. So this is a theta-like structure of DNA. So how exactly prokaryotic DNA replicates? So there is a replicon model. Okay, the replication begins at a specific initiation point, also known as origin of replication or replicon. In this image, you can see there is a circle, and there is a specific genes. There are specific gene present, which denotes as a initiator gene. And by following some of the genes or some of the nucleotides, there is a replicator nucleotides. There might be a 200 or 300 nucleotides which act as a replicator for the replicon model. It is a unit of genome in which DNA is replicated. It contains an origin of initiation of replication. It is a point of DNA open up and form open complex leading to the formation of pre-priming complex to initiate a replication process. So how exactly replication process get passed away? Then we will see. Now there are n number, like there are a bunch of proteins or bunch of enzymes which are as we know that enzymes are nothing but proteins. So many of the proteins and enzymes are needful for this replication or throughout replication process. So how many enzymes and proteins are there? We will see that. So first is DNA polymerase. There are various types of DNA polymerases, but what exactly DNA polymerase and how it works, we will see. What exactly the function of DNA polymerase? It matches the correct nucleotides, then joins, polymerizes adjacent nucleotides to each other. So this is the basic function of DNA polymerase. Now there are three types: DNA polymerase 1, 2, and 3, and their classification is also present. Now the second is helicase. The function of helicase is to unwind the DNA and melts it. Okay, to separate out or to form a new copy, first step is to separate out parental DNA, upon which the new strand can be recreated. Now the primates provides an RNA primer to start polymerization. Now SSBP is nothing but single-stranded binding protein, which keeps the DNA single-stranded after it has been melted by helicase. Now the gyrase. Gyrase is also known as, or gyrase and topoisomerase having the same function. So gyrase and topoisomerase that relieves the torsional strain in the DNA molecule. After the functioning of topoisomerase and gyrase, there is a final stage. Okay, we will see. The last enzyme would be ligase. So ligase is nothing but molecular gum or molecular sticker, which joins two nucleotides which are breaked, or some of the nucleotides may be accessed out or may be inserted or may be deleted. That site must be ligated by using ligase enzyme. Now telomerase, which finishes off the end of DNA strand in eukaryotes, which is specific for the eukaryotes. We will be not seeing this in prokaryotic replication. Now, how many enzymes and proteins of DNA replications are present in this prokaryotic replication? Now, there are many proteins like DNA A, DNA B, C, G, SSBP. We will see their functions simultaneously. Now what is a DNA A protein and how many subunits and molecular weight there is present in this chart? You can see. And basic function of DNA A is to recognize ORIC. ORIC is the site where the replication starts. Okay, this is a particular site where some nucleotides are present, which signify this is a particular site where you have to where the replication should be started. Now the DNA B protein, which is also known as a helicase protein or helicase enzyme. The function of helicase enzyme is unwinds or open the double-stranded DNA. Now, DNase protein. Actually, DNase protein not having that much any important word, but it supports DNA B to unwinds and open the double-stranded DNA. Now, DNA polymerases, which synthesizes the new DNA strand, and which is also present for the proofreading activity. Now DNA G protein, which having one subunit, and the function is synthesizes RNA primer. Now single-stranded binding protein SSBP, which having 4 subunits, and the function is to bind single-stranded DNA. Now DNA gyrase having 4 subunits, and which relieves the torsion strain from the DNA. Now we will see the types of DNA polymerases and their functions. Now, the first type is DNA polymerase 1, which was firstly invented. The first DNA-dependent DNA polymerase, which is also known as DNA poly 1, was discovered in 1958 by Arthur Kornberg, who received Nobel Prize in Physiology and Medicine in 1959. DNA polymerase is also considered or also known as Kornberg enzyme. Later, DNA poly 2 and DNA poly 3 were identified. All of them possesses the following biological activity. Now, first is 5' to 3' direction. Every nucleotide is added only from the direction 5' to 3' direction. Okay, because of the OH group or the phosphate group is only present at the 3rd position of the carbon ring. Okay, so the nucleotides added by only the 5' to 3' direction. It is also known as polymerase activity. And the second activity is exonuclease activity. Now, what is exactly the exonuclease activity is? Exonuclease activity must be from 5 prime to 3 prime and 3 prime to 5 prime. When 5 prime to 3 prime activity happens, the function of exonuclease activity is to remove primers or excise mutated segments. And when the exonuclease activity is carried out from the 3 prime to 5 prime direction, the main function of is to excise mismatched nucleotides. If any of the mismatched nucleotide will be there, then it would be excised out and then relocated by the new nucleotides. Now the function of DNA polymerase 1, mainly responsible for proofreading and filling the gaps, repairing DNA damage. If the DNA damage happens at that time, DNA polymerase 1 is useful. Now in DNA, there is a N-terminal and C-terminal. When the DNA poly 1 get when form the cut, there is a small fragment and large fragment. Small fragment having 322 amino acids having 5 prime to 3 prime exonuclease activity, and large fragment having 604 amino acids, and which is also known as clean off fragments, having DNA polymerization and having 3 prime to 5 prime exonuclease activity. Now DNA polymerase 2, the function of DNA polymerase we will see. Temporarily functional, which is temporarily functional when DNA poly 1 and DNA poly 3 are not functional. At that time, DNA poly 2 plays an important role. Still, which is capable for doing synthesis on the damaged template, which participates in DNA repair process. Now, DNA polymerase 3, which is also known as holoenzyme or a heterodimer enzyme, which is composed of 10 different subunits. Okay, so there must be alpha, beta, gamma, delta, delta prime, phi, psi. We will see step by step. You will get it soon. Okay. Yeah, the a heterodimer enzyme composed of 10 different subunits, having the highest polymerization activity, which is 10 raised to 5 nucleotide per minute. So high efficiency of DNA polymerase 3 we can see. The true enzyme responsible for the elongation process. This is the actual enzyme which is actually responsible for the elongation of nucleotide process, replication process. Now, there are 10 holoenzymes. I have just tell you. There are 10 enzymes. There are 10 enzymes: alpha, epsilon, theta, tau, gamma, delta, delta prime, phi, psi, and beta. How many holoenzymes it contains, their molecular weight, and how their gene, how they signify their gene and their functional subunits. So alpha, epsilon, and theta, which are basically useful for the polymerization activity. Okay, so polymerization activity is only done by the 5 prime to 3 prime direction. Okay, and when proofreading activity should be there, so proofreading activity is only happen when 3 prime to 5 prime activity is done. So these are the enzymes which is also known as core enzyme. Now the tau, gamma, delta, and delta prime, these are the clamp loader systems, and which is useful in the elongation process. Now gamma and epsilon and beta, which are helpful in interaction with single stranded binding protein, which is also useful for the clamp loader subsystems. Now, structure of DNA polymerase 3. DNA polymerase 3 contains 10 different enzymes which are useful for the DNA polymerization reaction. So what does alpha does? Okay, which having 5 prime to 3 prime polymerization activity. Now epsilon has 3 prime to 5 prime exonuclease activity and plays a key role to ensure the replication fidelity. Now theta maintains heterodimer structure. Nucleotides are always added to the growing strand at the 3' end because the new nucleotides are added only at the 3' end OH hydroxyl group, free 3 OH group. At that position, only the new nucleotides get added. So the end at which the DNA strand has a free OH group on the 3' carbon of its terminal deoxyribose. Now, what exactly the RNA prime is? It is also known as DNA gene. Primase is able to synthesize primers using free NTPs as the substrate and the single-stranded DNA as the template. Primers are the short RNA fragments of several nucleotides long. Actually, RNA primers are nothing but the molecular engines. How engine works? Okay, engine pull up the entire train to get run out. Okay, so primer is a main engine in the polymerization activity. Now this is a DNA template 3' to 5', and when remember one thing, as I have told earlier, the new nucleotide only added on the hydroxyl group, right? So hydroxyl group is present at the 3' end. So 3' end should be the free end. So for the RNA primer, it added 5' to 3' direction. RNA should be a long nucleotide, a short segment of RNA, because this is a single-stranded DNA, single-stranded RNA, right? DNA is also double-stranded and RNA is single-stranded. So just to form a new template, there is a need of single strand. So it plays an important role for the polymerization activity. When the RNA primer add by using template DNA or parental DNA, it elongates the chain by adding new nucleotides. Primer provide free 3-OH groups to react with the alpha phosphate atom of DNTP to form phosphodiester bonds. Primates DNA-B, DNA-C, and the origin form a primosome complex at the initiation phase. Now the function of helicase. Helicase is to separate the two strands. Okay, so it is also known as DNA-B. It opens the double strand of DNA which consuming ATP to break out this what can I say, like bonds which are present in two nucleotides or two strands of DNA. There must be a use of ATP to gain the energy. When the energy gains, it removes or it melts the bonds which are present in the two strands. The opening process with the assistance of DNA and DNA-C. Now, what exactly the single-stranded binding protein? Single-strand DNA binding protein maintains the DNA template in the single strand form in order to prevent the double-stranded DNA formation and to protect the vulnerable single-stranded DNA from nucleases. Now, what is DNA gyrase? This is the main enzyme which plays important role. Along with the DNA polymerase, DNA gyrase is also an important enzyme or protein. It cuts the phosphodiester bond on the both strands of double-stranded DNA, which releases the supercoiled constraint and reforms the phosphodiester bond. It can change a double-stranded DNA into a negative supercoiled state with consumption of ATP. To cut the phosphodiester bond, there should be a AT-rich sequence. Okay, so to break out the sequence, there must be a energy required. So to gain the energy, they conserve ATP. Now, how exactly DNA ligase works? DNA ligase is nothing but molecular gum or molecular sticker. So to stick out the two cutted DNA strands or two separated strands to rejoin them, DNA ligase is present. So in this diagram, you can see in colored diagram, there is a cut between two strands. So when the DNA ligase is get added to the particular strand, then two DNA strands or the separated strands get rejoined or reconciled, which connects two adjacent single-stranded DNA strands by joining the 3' OH group of one DNA strand to the 5' phosphate of another DNA strand, sealing the nick in the process of replication, repairing, recombination, and splicing out. Now, what exactly the proofreading and correction process? So, for the proofreading activity, there is a DNA poly 1 enzyme which has the function to correct the mismatched nucleotide. If mismatched nucleotide happens, it excises out that nucleotide and relocate the new nucleotide. It identifies the mismatched nucleotide, removes it by using 3' to 5' exonuclease activity, and add correct base and continues the replication process. Now, there are three main stages which are which are important in DNA replication process. Actually, these are not specific stages, but for our concept clarification, we just name for that. So there are three steps: initiation, elongation, and termination. So what exactly initiation? It occurs at the origin of replication, separate double-stranded DNA, and primer synthesis. Now, in elongation, involves the addition of new nucleotides, DNTPs, based on complementary of the template strand, which forms phosphodiester bond and correct the mismatch bases, extending the DNA strand. Now, final stage is termination, which stops the DNA replication, occurs at a specific site, which is also known as termination site. So, what exactly this diagram shows? We have taken E. coli, Escherichia coli example for the prokaryotic replication. In this diagram, there are many sites, okay, from which many functions can be carried out. For replication process, ORIC, origin of replication, is for the particular site where from the replication is started. So there is a ORIC point. There are some nucleotide presence, bases are present, which shows or which signify the site for the origin of replication. Now, the initiation process. The replication start at a particular point called origin of replication or ORI site. The structure of the origin is 248 base pair long and 80 rich sequence. Why 80 rich sequence is present? Because there is no need of high energy consumption. If we take GC content is high, then there must be a high energy must be required. So when 80 rich sequence is get, at that time they just search. If replication processes happens, they search for 80 rich sequence. When 80 rich sequence happens, at that time they start to cut out that sequence. Okay, in this diagram we can see in right side there is a 80 rich consensus sequences, and in left side there is a consensus sequences which having 13 base pair sequences. In the right side, there the DNA A protein binds. DNA, the function of DNA A is to find the origin of replication site, which finds that particular site and get attached to that the particular site. When the 80 sequence finds by the DNA A protein, there are these sequences are known as 9-mer sequences, and when the ending of that sequences, there is a 13-mer sequences, the ending of that origin of replication site. Now, the formation of pre-primosome. In this diagram, we can see there is a 9-mer sequences. Okay, the 9-mer sequences are highly applicable for the applicable for the replication origin of replication site, and 13-mer where the origin of replication get completely covered up. When the DNA finds that particular 9-mer sequences or 80 rich sequences, it binds to that sequence and ATP energy get accessed out to break the particular site, because to cut the particular sequences or particular bases, there must be a need of energy. So for energy, ATP required. So for the breaking of nucleotides or breaking of bonds, there is a need of DNA B, also known as helicase. So to help the DNA B or helicase, DNA C also helpful. So this is all compensatory known as chromosome complex. Now origin of replication, where the site of DNA synthesis start. In this diagram, you can see there is a particular point. This is the origin of replication. From that point, you can see there must be a replication process actually starts. There are some initiator proteins which initiates the replication process. There are must be some bases or some of the nucleotides which helps to initiate or to help to originate the sequence, or to help to DNA protein to get attached to that particular point and to follow the furthermore cycle. Formation of replication 4. Basic step is to DNA. DNA enzyme recognizes the particular site, that is ORIC. When it recognizes the ORIC, the main function of DNAB, the DNAB function will do. DNAB function is to cut the AT-rich sequence and separate out the two strands. Okay, so DNA A and DNA C join the DNA A complex, open the local AT-rich region and move on the template downstream further to separate enough space. DNA A is replaced gradually. SSB protein binds to the complex to stabilize the single-stranded binding protein. When DNA separate out two strands, to hold the one strand, there is a need of any of the protein. So at that time, single-stranded binding protein plays an main role. Okay, to bind that particular strand or bind a single strand, SSBP protein plays important role. Now primer synthesis. Primates join and forms a complex called primosome. Primates start the synthesis of primers on the single-stranded DNA template using NTP, nucleotide triphosphate, as the substrates in the 5' to 3' direction at the expense of ATP. The short RNA fragments provide free 3-OH groups for the DNA elongation. As we all know that 3-OH group must be free for the new nucleotide add. Now in this diagram, we can see there is a Y-like structure. So from 5' to 3' direction, as we know that when DNA polymerase activity only happens when the 5' to 3' direction is there. So in the leading strand, there is a 5' to 3' direction. So when how primer works? Primer is, we know that primer is an engine, nothing but the engine. We just provide somehow sequences to elongate the furthermore nucleotides. So in leading strand, only one primer is needful, but at the lagging strand is 3' to 5'. So at the 5' end, it can't be add new nucleotide, because new nucleotide can be added only at the 3' end free 3' end group. So at that time, when 5' OH group get it to us, at that time primer continuously added. When one primer is added, it will help to elongate the some nucleotides, but after that it will stop the polymerization reaction. Then again another primer is added, and continuously the process will be happen. So in the leading strand, only one primer will be required, but at the lagging strand, there must be n number of primers would be required. DNTPs are continuously connected to the primer on the nascent DNA chain by DNA polymerase 3. The core enzyme alpha, epsilon, and theta catalyze the synthesis of leading and lagging strands respectively. The nature of the chain elongation is the series formation of the phosphodiester bonds. So in the previous diagram, we have discussed, okay. So then, Okazaki fragments are there. I told that there are n number of primers. When proofreading activity happens, that primers get excised out. When that primer get excised out, there must be some filling nucleotides would be there. there must be a addition of new nucleotide at that time. They form a small gaps. That gaps are nothing but Okazaki fragments, and then that fragments get ligate or gain that what can I say, get
joined by
connected.
yes.
Connected, yeah, joined by ligase enzyme. Now, lagging strand synthesis. RNA primers on Okazaki fragments are digested by the enzyme RNase. The gaps are filled by DNA poly 1 in the 5' to 3' direction. The nick between 5' end of one fragment and 3' end of the next fragment is sealed by ligase. Now, what exactly Okazaki fragments? Many DNA fragments are synthesized sequentially on the DNA template strand having the 5' end. These DNA fragments are called Okazaki fragments. They are 1000 to 2000 nucleotide long in prokaryotes and 100 to 150 nucleotide long in eukaryotes. The daughter strand consisting of Okazaki fragments is called lagging strand. So there are many primers are there. So many Okazaki fragments would be present. So on the lagging strand, there are Okazaki fragments are present. So directionality of DNA strands at the replication fork. As we all know that in prokaryotes, there is a bidirectional replication process happens. So in lagging strand is carried out from the 5' to 3' direction. In leading strand, it happens from 3' to 5' direction. So at a particular point, it must be reconciled. So this is a site where bidirectionality of replication we can see. Uh, in this diagram, how the proteins works we can see. There are two strands, 5' direction, 5' to 3' and 3' to 5'. 5' to 3' is leading strand and 3' to 5' is lagging strand. So to uh, to separate out the DNA, to separate out two strands, there is need of DNA B protein or helicase protein to hold out one strand. To hold out one strand, there is a need of single stranded binding protein. Okay, when one strand connected by the single stranded binding protein, then RNA primer get added to a particular sequence for the 3' to 5' direction. RNA primer get added, and then from the 3' direction, new nucleotides get added by the DNA poly 1 or DNA poly 3. At the termination process, the replication of E. coli is bidirectional from one origin, and the two replication forks must be meet at one point called ter at 32 nucleotide long. All the primers will be removed and the fragments will be connected by DNA poly 1 and ligase. Now, actually, termination of DNA replication occurs when two oppositely oriented replication forks meet and to fuse to create two separate or complete double-stranded DNA molecules. The termination process actually happens. In specific to our prokaryotic replication, the particular region which is restricted to the termination process, which is also known as termination region, and which might be present at the exactly opposite to the origin of replication site, where the origin of replication start, and at the opposite exactly opposite it is termination process. So for the termination process, there is a need of ter proteins. So ter proteins are which is responsible to get out two strands rings separately. So ter proteins are binding site for the TUS protein. So what exactly TUS proteins? So TUS proteins are terminus utilization substances, are nothing but the proteins or enzymes. So in this given diagram, we can see the termination sites like A, B, C, D, E, F, G, H. No, only F. Okay. So only G are found to present on the DNA. So on this sites, termination A site we can see terminates the counterclockwise direction. So from the termination ter A, from the ter A side, it goes from clockwise direction, and from the ter C, it goes from its clockwise direction. So TUS proteins bind with the TER to form a TUS and TER complex, and thus the both bidirectional fork get then finally get arrested by one direction and meet up at the particular point. And that particular point is known as DNA knotting. And that DNA knotting, like example, we can take an example. If one bangle, hatatli bangdi, uh, if one bangle is get mingled into another bangle, then how can we remove it? By just knicking out, or by just moving a small gap, and then we can remove out that another bangle. So at the same phenomena, this is happens here. So, and the finally, two progeny duplex DNA synthesis, the synthesis can be done. So, two separate DNA strands of prokaryotic DNA get happens. Okay, thank you.
Thank you, Aditi Deshpande madam, for the presentation on prokaryotic DNA replication. You have elaborated all the aspects of DNA replication. And being, I am also a zoologist. I also teach the molecular biology. And I have my specialization molecular biology in my PG. I also did my PG.
Oh, wow.
Yes. So I aware about all this topic, and my student might be have understand this topic. It's quite difficult to understand for the UG life science student the prokaryotic DNA replication. But when they get concentrated on the topic, they will get definitely understand the topic.
I also thought that actually this is a long or vast subject, so it is somehow difficult to get get completed in 1 hour. So I asked to Manish Deshpande to how could I complete in only 1 hour. So at my UG level, I have completed this in 8 days. So how could I complete in only 1 hour? So I took effort, and I just just tried my best.
You have precise. Yes, you have precise the topic and within hours. I think that I thought that it is only in 50 minutes you have completed your topic.
Thank you so much.
Yes, you have started at 11:10 and you have finished at 12, right? So you have precised it very neatly, and I hope yes, and I hope these two participants will understand the topic. If the participant have any query, they can ask, ma'am. Do students have any query about this topic? I thought that they have clear all the ideas, but if they have such kind of any difficulty, you can tune our YouTube channel, DBC Media Center, where you can ask your queries in chat box. So definitely either Deshpande madam or from our faculty members, any or me, resolve your queries. Definitely. Aditi Deshpande madam, once again from our institute, our president, Dr. Madhurav Patil sir, our principal, Dr. Chavan sir, convener of this conference, myself, Dr. Arvind Chauhan, co-convener, Dr. Bhupesh Nemaniwar, and all my faculty members. We once again say thanks to you for covering the such a either if you do not cover this topic in this CWCP, it will remain again because no one has a take chance to go for lecture on this topic, because either of the in the microbiology or in biophysics we have such this topic of prokaryotic DNA replication, but either in zoology we do not have a topic because this paper is not related to or any genetics paper also do not have. They have only the DNA replication. It's simply DNA replication, not a prokaryotic or eukaryotic. Yeah, so you have covered this topic in this CWCP. For that, from student, I thank you once again, and say greeting to Dr. Manish Deshpande sir. Yeah, Thank you. Thank you very much. The student, we will send a feedback link on today's topic. And just give us 2 to 3 minutes to give your valuable feedback on today's topic. Thank you. Thank you very much. For today, all the sessions are over here. We will tune at morning 10 o'clock with another topic, with another resource person. Thank you. Thank you very much. Thank you, Deshpande madam. See you again with another new topic if you have a time. Ma'am, unmute yourself.
Hello. Yeah, am I
Yes.
audible?
Yes, sir.
Yeah, thank you very much, sir, to giving me such a good opportunity. So actually, previously, I haven't worked over any of the platform, but this is a good platform where I can serve my good service to you, and thank you
Yes,
once
yes,
again.
exactly.
And
we have taken inspiration from Manish Deshpande sir that 1 year before he has took such kind of event for the NSB
yeah,
college.
yeah.
Yes, yes.
Yes,
From
yes,
that
yes.
event, we also take this opportunity, this motivation to run such a kind of activity for our student, and we are continuing this activity in May and onward. As per
Wow, wow, wow.
yes, yes, we are continuing because this platform will available student different kind of teacher from the either we can say it is from the all over the world. So we are inviting
Right.
some international guest faculty on some of the topic relevant to the students. We are trying to do. We are in communication with them. So definitely in a 1 month, one or two international faculty should be invited for their lecture on this platform. So we are trying to continue if we have a good support
Wow,
from
wow.
faculty and student also, because
No, no.
we are doing
Yeah,
this
yeah.
activity for the student, and if the student will not be there, then it will be like a waste for everything. But it will the lecture will be available on YouTube channel also. So whenever the student
Okay,
need,
sir.
they will assist there. Yes. Thank you,
No, no,
ma'am.
sir.
Thank you.
Okay,
Thank you very
no
much,
problem.
ma'am. If we,
Okay.
I think that you have molecular biology specialization in microbiology. Do
No, no,
you
no.
have?
Actually, I have done my master's in. No, no, no. Actually, I have done my master's in bioinformatics from
Okay.
RJIGBT Pune.
Okay. Okay.
So, not a specific microbiologist or molecular biologist. I am just a bioinformatician. But
Mm-hmm.
one of my from my curriculum, there were a subject like molecular biology. So, it was very much familiar to me,
Mm-hmm.
and it was my favorite subject during my
Okay. Okay. No issue. Then we will have a lecture on bioinformatics also because we have one special
Okay,
paper
sir.
on the
It would,
bioinformatics too.
yeah, yeah.
Yes, yes, yes.
Okay, sir. Please let me know if
And then
any
yes,
help would be there.
yes, yes,
I will be there, sir.
yes, sir. Yes, yes. BSc TY student and BSc TY
Yeah,
zoology
yeah.
student as well as biophysics. There we have bioinformatics.
Biotechnology
Yes,
students also having a chapter for one subject for the bioinformatics, I guess.
yes.
So,
Exactly. So definitely, I will let you know which topic you have to be chosen for this event,
yes, sir.
right?
Yes, definitely, definitely,
I
definitely.
will, yes, I will, yes. Yeah, I will
Okay.
communicate with Deshpande and yeah. Okay.
Okay, sir.
Thank you
Yeah,
very
yeah.
much, ma'am.
Thank you.
Just say
Thank you,
or
sir.
credit to Deshpande, sir. Yeah.
Okay, sir.
Thank
Okay.
you.
Fine. Thank you.
Yeah. See.